Yeah, so I'm recording it if that's okay with everyone else. If you have any problems, you can type it in the chat and I can stop recording as soon as you tell me. Okay, so hi again, my name is Natalia and I'm the co-president of AMPC alongside Adam Snyder. Oh, we have someone else joining us. Thank you so much for coming to AMPC's first ever research symposium. So the symposium started in late November or early December of last year with the purpose of allowing teams of students to gain a deeper understanding of and more experience involving research. So by doing so, and although these have been some challenging times, the students are able to further develop their teamwork, problem solving, networking, writing, leadership, interpersonal, and analytical skills. We were fortunate enough to be joined by exceptional researchers in many different medical fields. And so we have more than 20 teams working on research on various health-related topics. For example, today we will have teams presenting on glioblastomas, neurodegenerative disorders, muscular dystrophy, cells of the nervous system, genomics and bioinformatics, and others. So I would like to thank you again for joining us today, and I am thrilled to see what the students have been working on the last few months. I will now allow Victoria to introduce herself and the first symposium teams as well.
Hi, everybody. My name is Victoria, and I'll be helping Natalia moderate the event. I'm on the e-board of AMPC, and I'm super excited to hear everybody present and see what kind of work you guys have been doing. The first team for today, Team 1, to start at 3:05, is Casey. You can share your screen, yeah.
Hello everyone, my name is Shukwan Marizuma, but most people call me Casey, and today my team and I are going to be presenting neurodegenerative and neurobehavioral diseases. Due to the pandemic, we use this opportunity to conduct a literature review by reading through a multitude of research papers to form the basis for our research topic, which is neurodegenerative and neurobehavioral diseases. We originally chose this topic because we were all majoring in the field of neuroscience and wanted to look into a topic that combined our interest in understanding the origins of neurodegenerative diseases and how it impacts behavior and the expression of genes. So neurodegenerative diseases, NDs. What are they? These are disorders such as diseases in which neurons and glia stop working or die off and progressively get worse as time goes by. Neurons and glia are cells that are found inside the nervous system. Neurodegenerative diseases/disorders have no cure, with causes ranging from genetics, aging, tumors, strokes, viruses, and in some rare cases, unhealthy lifestyles. These unhealthy lifestyles can include anything from excessive alcohol consumption to other toxin consumptions. So in this slide, we're going to be going over certain triggers of neurodegenerative diseases. Idiopathic NDs result from the combined actions of multiple risk factors that include genetic predisposition, age, and environmental triggers such as toxins and inflammatory responses involving microglia and astrocytes. In one such scenario, common viral infections can trigger peripheral local inflammatory responses, which lead to early neuronal and glia dysfunction in the CNS. Peripheral inflammatory reaction can also initiate neurodegenerative changes in the PNS, which are often associated with the preclinical phase of many neurodegenerative diseases. Such induced acute or chronic inflammation involving cytokines and activated glial cells could therefore be responsible for the priming of PNS and CNS to degeneration by reducing the threshold for irreversible neuronal damage resulting from any subsequent neurotoxic trigger. Genetic susceptibility to neurodegeneration can also accelerate neuronal damage in response to inflammatory triggers. Viral invasions and inflammation. Viruses can trigger neuronal dysfunction by direct cytoclic effect, direct neuronal priming, or by standard inflammatory reactions. Neurodegenerative diseases, NDs, including Alzheimer's disease and Parkinson's disease, are a group of debilitating disorders of the central nervous system affecting memory, reasoning, emotions, and motor skill at the cellular level. Damage in neurites, axons, and synapses, brain inflammation, and loss of neurons underlines the pathology. Currently, there's no effective treatment for NDs. The prevalence of ND significantly increases with age. As the global population is aging, NDs are predicted to cause severe social and economic burden. Research in the last 50 years has provided insight about the role of genetics, environment, protein misfolding, protein homeostasis, oxidative damage, neuroinflammation, and the pathology of NDs. Many of these apparently diverse mechanisms appear to converge into mitochondrial maintenance and function as a key aspect of the disease pathogenesis.
Hello, everyone. My name is Wasif Khan. I'll be talking about the neuroinvasion mechanisms of a particular virus, SARS-CoV-2. Of course, it's a pretty prevalent virus that we're all aware of. SARS-CoV-2, it's short for severe acute respiratory syndrome, and a common receptor protein that it binds with is called angiotensin converting enzyme 2, also known as the ACE2 receptor. And like Casey said earlier, neurodegenerative diseases can be caused by viruses, and the virus that I'm talking about, of course, is SARS-CoV-2. Next slide, please. So this diagram represents proposed mechanisms that SARS-CoV-2 can embark on, and there's still ongoing research to further support its validity. These mechanisms are based on previous human coronaviruses and the mechanisms that are proposed for them. However, they aren't solely the only mechanisms that are out there, as there are also other mechanisms that do come to mind. But because that this mechanism of the neuronal retrograde spread and a hematogenous spread, they have a lot more research from previous human coronaviruses. That's why I decided to stick with something where there's more information of. But of course, it's not the only pathway that exists. Next slide, please. So yeah, one of the mechanisms from the previous slide I want to talk about is the hematogenous pathway, which is where SARS-CoV-2 particles could potentially infect endothelial cells of the blood-brain barrier in order to enter into the central nervous system through the virus's inducing effect on a cytokine storm. And of course, too many pro-inflammatory cytokines can be potentially harmful for our host cells, which, by the way, the blood-brain barrier, of the endothelial cells, it's a semi-permeable membrane that limits, let's say, access of large pathogens into the cell and therefore the central nervous system. Cytokines of one kind could induce various types, such as if we look on the right, there's a angiotensin 2 or the ANG2 cytokine. It's through multiple pathways. It's influencing the rise of other cytokines, such as the IL-6 cytokine, and along with various other chemokines through, again, multiple pathway mechanisms. And it is by cytokine storm that patients can experience neurological manifestations such as acute respiratory distress syndrome that's been observed in COVID-19 patients from the articles that I've read. Next slide, please. Another mechanism is the neuronal retrograde pathway, which is where viral particles can attack the neurons in the peripheral nervous system and travel to the central nervous system through an axon transport. Viruses tend to reach the nervous system through the olfactory pathway, where the axons of these neurons stretch from the nasal cavity and all the way up to around the olfactory bulb of the brain, which is a pretty common route. And if we want to connect this mechanism with our common understanding of SARS-CoV-2, I mean, if we think about it, we're wearing masks and to cover our noses, and if the virus particles were to enter up through our nose, it would thus really exhibit this neuronal retrograde pathway. But again, this isn't the only pathway. There are others as well. Next slide, please.
Hello, my name is Ankita Sahai, and in relation to the research paper, I examined the relationship between neurodegenerative diseases, or NDs, and behavior with a focus on viruses and their impacts on the host body. Next slide, please. So I looked at an article that discussed the rabies virus, which is a deadly virus that's most likely to be spread through the saliva of infected animals in the context of gene expression. So the rabies virus can also impact gene expression in the host's brain by activating approximately 39 genes through infection. Despite that expression, 16% of the represented genes in the experiment were expressed at significantly higher levels in a normal brain as compared to an infected brain. This indicates that the rabies virus can also underexpress certain genes. Along with that, further testing comparing the brains of uninfected and infected brains revealed 2,688 differentially expressed gene fragments which support the significant difference in gene expression discussed previously. It's also expected that the replication and spread of rabies depends on a sequence of host gene functions that parallel the replication of rabies proteins and subsequently the production of rabies. Despite this research, the impact of viral illnesses on gene expression is complex because there are several effects that can occur depending on each virus strain. Next slide, please. So next I'm going to talk about genes and the expression of language. Casey previously discussed Parkinson's disease, which is an ND with some genetic basis that most greatly impacts the central nervous system, specifically movement. The genetic basis is complicated because it's determined by a number of different disease-causing and disease-predisposing genes. Communication impairment is also common in this disease, present in up to 90% of patients. Such impairment may have a basis in both motor speech control and cognitive linguistics. The severity of communication impairment varies between individuals, but it can progress to a lack of functional communication. Now, functional communication is the way in which individuals spontaneously and casually communicate their thoughts. Functional communication is a crucial concept to understand because it differs from the communication defects that we observe in a controlled research setting. Since Parkinson's disease impacts both motor and cognitive function, it's ambiguous as to which communication defects are based on effects on motor speech control and which have a cognitive or linguistic basis. Next slide, please. Lastly, I wanted to briefly touch on the psychopathological symptoms and impacts of various NDs. Many NDs can result in disruptions in emotional, cognitive, and social behavior, largely in part due to the loss of neurons. This graph specifically looks at different symptoms of psychopathology in relation to Alzheimer's disease. While social behavior is often preserved and relationships can be maintained as the disease progresses, we can see that the prevalence rate for apathy, anxiety, and dysphoria tend to increase with the severity of the disease. This is all to emphasize that NDs undeniably impact the emotional realm of an individual along with their cognitive processes. Next slide, please. So this is the researcher that we worked with, Dr. Eleftherianos, who unfortunately had an emergency to attend to. Dr. Eleftherianos' research focuses primarily on host pathogen interactions using a model insect. So in his words, previously we have performed a significant amount of work to extend our current understanding of the signaling events that control the immunopathological defects of virus infection in the Drosophila model. Here we research the types of neurodegenerative diseases, the relationship between virus infection of the nervous system and neurodegeneration, virus-induced neurobehavioral effects, and lessons that we can learn from small model organisms for dissecting virus disease and brain function. This information is important to appreciate the complex processes that regulate the host, antiviral, immune, and nervous systems, and therefore it will be important to neurobiologists, immunologists, virologists, microbiologists, and physiopathologists, and to those studying the neural basis of host-virus interactions. The next slide is references, but we'd like to open the floor to any questions that anyone may have. If there are no questions at this exact moment, we can actually talk about our own process researching and studying these articles. Wasif, if you'd like to start, you can go for it.
Sure, yeah. So for us, for our literature review, our future goal is to publish it, of course. Once we get back with Dr. Eleftherianos, we'll try to fully analyze everything we've worked on of our literature review and hopefully get to the publication process as soon as we can. In terms of how we managed to dissect creating the literature review paper, but we first met back in, I think, January or so. We wanted to come up with a topic, and because me, Casey, and Ankita, we plan on having a neuroscience major, we knew that we wanted to have that as our focus. So, and after a couple of weeks of planning what exactly we wanted to spend many months on creating a paper out of, we decided to go with our topic of neurodegenerative diseases and neuroinvasion pathways. And in particular, when we met in February, we also decided how myself, Ankita, and Casey, and Dr. Ilif we would work on our own portions of the paper and then combine them, which the combination process, of course, will begin next week. And in terms of what we've learned as a whole, I believe that I'm sure that this was the first time for myself, along with Ankita and Casey, working on a vigorous project like this. And it was a fun process at the same time of reading the various sources that were out there and the multiple databases we looked at, getting multiple perspectives on the topic of neuroinvasion or just neurodegeneration in general. We don't know the totality of it. We don't have PhDs, but we hopefully have enough background knowledge to further support our future education in regards to the classes we take in our future pre-health careers.
Going off of what Lassie said, I just definitely learned a lot throughout this entire process, specifically when it comes to the overview of neurodegenerative diseases and disorders. Working with them has definitely showed me that with like-minded people, you can achieve great things.
And something that, to mirror what Casey and Wasif have said, and something that I really enjoyed in this process was really understanding the impacts of neurodegenerative diseases on language, because understanding how our brain processes language is something that I'm personally extremely fascinated by. So I think being able to do this literature review helped me further understand all those neural processes.
And yeah, that's what we.
Yeah, thank you for paying attention to our presentation.
Thank you all for listening.
Thank you all so much. I was actually going to ask you earlier on about what we had learned, so it was good that you touched upon it before I ended up jumping in. But I was wondering, thank you so much for sharing with us everything that you've been doing for the last few months and how much you've learned so far. What was your favorite part of doing this research? I know that to conduct a literature review requires much patience and also teamwork skills. And so I was wondering, what was the best part for you in this different journey or experience that you had?
For me, I would definitely say being able to meet with a professor that specializes in some type of neurodegenerative disease, especially when it comes to animal-like models like Drosophila. So being able to talk to someone about something that I plan on majoring in really did open my eyes to like the possibilities that I could potentially go down like after I graduate.
I think, yeah, for the best part for me was really the journey itself, not necessarily the product, because it wasn't easy. We're looking at various articles, having a basic understanding of what we're reading and even much more of a better understanding. And I think once we got to each of us, once I got to the point of like understanding, okay, I think I have a good knowledge of what I'm reading, and then I thus began writing some stuff like drafts of my research, I thus felt very good about what I was able to accomplish. And the fact that I managed to do that my first time around gives me enough confidence to know that I could continue to work on future literature reviews if I choose to embark on that. So the journey itself was very fun for me.
Yeah, I agree. I'm also just so fascinated by, like, as I said earlier, how we process language and how we learn it. So I think really seeing all that come together and how that really manifests itself in the context of neurodegenerative diseases was extremely eye-opening for me, because like most of what's in my head about it is like theory, but just seeing all the research behind it was super cool.
It's amazing. Thank you so much. Okay, so going forwards, the next team is led by Dr. Lau Chalupa, and it's composed of the following students. It's Ria Varghese, Mishkat Habib, Victoria Rainey's, and Monica Kapoor. I believe we don't have all of them here today, so they asked me to share with you a pre-recorded video of theirs, but we do have some of the students here to answer any questions that you might have. So I'll be sharing my screen now and hopefully it works out. So here we go.
Hey guys, so me and my teammates have done a research paper on glioblastomas. We talked about the history of glioblastomas, the biology, and where the field is heading in terms of current treatments and clinical trials. It's done by me, Ria Burgess, Mishkat Habib, Monica Kapoor, and Victoria Reynes.
So the overview of the paper. So our goal was to propose a literature review, which includes an overview of the past, present, and future glioblastoma treatments, and also the history of classification. The goal was to use this paper to better understand where there are gaps in the field and to see where we can fill in those gaps. The topic is glioblastoma treatments and classification from when brain tumors were just discovered to modern day clinical trials. And we are currently in the final editing stages and looking to get this paper published.
So now we're just going to go through the outline of the paper. So first I'm going to talk about how we gathered the data. So we looked up a lot of research papers and articles on the Gelman library and also on Google Scholar. And then we divided, because there's so much of information about glioblastomas, we divided it into the history, the biology, current treatments, and where the field is heading, and so that we could divide the work equally. And then we also color-coded the articles so that when we put it in the paper, so that it's like, you know how to cite it. And then we put everything into a draft, and that was our first draft. And then we revised it, and Mishkat's going to talk about that. So this is how we wrote the first draft of our literature review. We divided it into 6 different sections, and all of these sections was divided between the 4 of us. So the introduction was done by Ria and Victoria, and then the history of glioblastoma was done by Ria and Victoria as well, along with the biology of glioblastoma. The section on current 2021 research on glioblastoma was done by Monica, and the clinical trials was done by me, and the conclusion was done by me and Monica. So each person researched and wrote the first draft on their own section, and then we came together and we read each other's sections and tried to combine it all and integrate it into one research paper. We also went through it together once on a Zoom call just to check if we had made any mistakes, and so everyone could suggest improvements. Then lastly, we sent it to Dr. Chalupa and to get his feedback. After we got his feedback, we made any necessary changes according to his advice and sent it back to him, and then he told us it was good to go. So we're still working on it, but I think we have it down pretty well.
Now we'll talk about what we have learned throughout this entire research process. So we focused on writing a literature review on glioblastoma. So we, of course, learned the process of writing a literature review, which encompasses gathering relevant sources such as scientific journals and analyzing them. We also learned that diagnosing glioblastoma and current research is very, very important to the field. And as such, there are traditional treatment options available, such as surgery, radiosurgery, and chemotherapy. However, new research in glioblastoma can enhance treatment options available for patients. Next, we'd like to expand more upon glioblastoma multiform. So glioblastoma multiform is known to be an aggressive tumor. In fact, it is classified as a grade 4 tumor. And new research and clinical trials are being conducted to increase efficient treatment options, which is one of the aspects we focused on our paper. And new treatments research in 2021 include new ways to model and diagnose glioblastoma, because diagnosing cancer as fast as possible is crucial for the patient. And researchers are also focusing on various surgical adjuncts to be able to deliver treatments that can pass through the blood-brain barrier. And lastly, researchers are also focusing on laser interstitial thermal therapy, which can target specific tumors to destroy. And these are just some of the various treatments we've come across for glioblastoma, but there are many others still being researched. So our future plans are to, of course, continue researching more into the field. We hope to gain more insight into glioblastoma research and various treatments, and we want to continue working on our literature review, and we'd like to add more information to the paper as new research in the field becomes available. So thank you. That's all we have for you today. We would like to answer any questions that you may have.
So I have a question. In your research, did you come across information about what are the driver mutations and which defects in which genes promote development of glioblastoma?
So we each took a part of the research paper to explore. So my other teammates, they were in charge of exploring the biology aspects of glioblastoma, but they're not here with us right now, unfortunately. So I can't answer that question. Unfortunately, I apologize.
Okay, no problem. Thank you.
I have a question. What treatment do you think is the most promising? Like, which novel treatment do you think is the most promising for glioblastomas?
I think just looking at all the various treatments, I think all of them can be promising because they're trying to build off of the more traditional treatments. But just going off of them, I think the medications that can pass through the blood-brain barrier seem promising because as of right now, a lot of medications can't pass through the blood-brain barrier to reach the glioblastoma tumor cells. So I think that's an interesting avenue to explore. And also another treatment is the use of different surgical adjuncts. So making treatments more safer and effective for the patient so that all the tumor cells and cancerous tissues can be removed as efficiently as possible. And another treatment, well, not really a treatment, but it's a diagnosing way is to model and to diagnose the glioblastoma earlier in a patient. So looking at how differentiating between cancerous tissue and healthy tissue, because a lot of times in an MRI scan or a CT scan, it can be difficult to sometimes distinguish a glioblastoma tumor if it's difficult to find in the scan. So looking at new ways to enhance the scans can be another fascinating way, I think.
Thank you.
Are there any other questions I can answer? If not, I hope you guys enjoyed our team's presentation. We hope to, of course, keep exploring the field of glioblastoma and finding new treatments that researchers are exploring, and we hope to keep updating our paper. Thank you so much.
Thanks, Monica. That was a wonderful presentation. Just wanted to say that. Thank you.
I'm just wondering if the next team, Paris's team, that's supposed to start at 3:35 is here. If you guys want to wait until 3:35, that's completely okay. But if you're all here and ready to present, you can also do that. So just let me know.
Yeah, we can pretty much start like whenever. I'm ready to go whenever.
All right, so if you would like to begin, the floor is yours. You can share your screen.
Okay, sounds good. All right, cool. Can you guys see it? Cool. Okay, so our research project focuses more so on the development of, it studies various dance styles, and it sort of focuses on which one creates the most enjoyable and the most effective workout. And our research team consists of me, Ananya, and then Dr. Todd Miller, who is an exercise science professor at Milken, the Milken School here at GW. So what's our research about? Our research mainly focuses on how we sort of talk about how exercise it has a lot of physical and mental health benefits, but at the same time, it's very hard to make a consistent habit of that because some people may not find muscle strengthening or cardiovascular exercises as exciting and therefore not as repeatable as other forms of exercise are. There have been studies conducted on how dancing has been shown to be a very repeatable and also efficient way of gaining exercise, of getting better at exercising and everything. And so our research focuses on these studies, and it discusses how there may be potential medical uses for combating the obesity epidemic and for promoting mental health as well through the development of a dance workout. And so our research question focuses on which type of dance style is the best form of exercise. And the best form of exercise in our experiment is defined as one that is highly enjoyable and also burns a large amount of calories. Similar studies like this have been conducted in the past, and they use similar experimental methods, and we took a look at those methods. And so this is a chart just to sort of bring home the point of how the obesity epidemic in the US has been increasing. And so therefore, this just shows how our study means to combat that and to sort of develop a workout that will help hope to reduce this epidemic in the future.
As for what we've done so far with this project, we've completed all the city training that is needed to perform the experiment and are in the process of working on IRB approval currently. And we've also completed an analysis of past studies that are similar to the one we're looking into exercise, exercise enjoyment, dance as exercise, etc., in order to help us design the most effective experiment, which we're going to talk about next, the methods that we've developed to outline what we're planning to do with our research.
Yeah, so as we mentioned for the methods for our experiment, we plan to use one edition of the game Just Dance. And the reason we're using this is because it offers a wide variety of dance styles that we can use to examine in this study. And so the way we're going to proceed about this, we're going to select three songs from the selection, and they're all going to have very, very different music styles. And this is so we can essentially sort of figure out which music style or which dance style is the most effective exercise in terms of our definition for this study. We're going to recruit 40 college students at random. We're going to measure their O2 consumption for a single song and give them some time to rest and catch their breath. And then they can come back another day and we can collect the data for later songs, because this will give a more accurate reading of the O2 consumption. Similar past studies that we examined, they measured the O2 consumption, blood lactate levels, and the heart rate of these dance workouts that were studied. For our study, we're going to focus on O2 consumption as it is very non-invasive, and we're going to use a device called a Quark CPET indirect calorimeter. And this device is going to calculate the calorie consumption based on O2, the calories burned based on O2 consumption. And the way it does this, it's going to calculate how much oxygen is consumed, and it's going to translate that into 1 liter of oxygen consumed gives you 5 burned calories. As for the enjoyment aspect of it, which we're going to measure, we're going to use a similar scale conducted in the past called the Physical Exercise Enjoyment Scale. This was also used in a past study. What this scale did was it presented a series of questions for the participants to gauge their enjoyment in the songs in. And so we're going to use this as well to measure the enjoyment. And we're going to combine the factors of calories burned, along with physical enjoyment in order to gauge what the most effective dance style is in this case.
What do we hope to learn from this investigation? We have our own favorite dance styles, but we're wondering if there's one that's universally liked. Additionally, can that style help create future workout plans that'll be more enjoyable and then easier to stick to exercise plans with? Additionally, can this study be applied to movement therapy, which was developed from dance, as well as it's been shown that dance workouts can be used in treatment of eating disorders and body image problems. So theoretically, the results of our experiment might be able to inform future therapies and treatments for those problems as well. Also, there's research that shows that there's dance stimulation machines that are used in Korean healthcare. And so it's possible that the results of our experiment could even be implemented into the US healthcare system by devising a more uniform, more applicable type of dance workout that could essentially be given to individuals. And
so what are the future plans for our experiment? So once we get the IRB approval, that's when we'll be able to begin the experiment itself. And so the study is planning to be conducted in Milken, and we're going to use that device that we mentioned before and we're going to perform the experiment there with the Just Dance game. In addition, we plan to compare the exercise benefits from each song from Just Dance. We're going to compare that to the recommendations from the American College of Sports Medicine. This was also done in a past study that we examined. And so this will help us create the best workout plan possible based on the dance style that is proven to be the most effective in this study. Right now, we're also examining mainly the physical benefits of a dance style, but we also plan to look into mental health benefits in the future as well, as exercise and dancing has been shown to have physical and mental benefits as well. In addition, Just Dance has a limited amount of dance styles that we can choose from, so we hope to expand this study later on to include other dance styles that are not included in Just Dance. And then finally, we are going to do something similar to what a previous study also examined. They looked at the effect difference in people with dance experience and people without dance experience. So we hope to do that in the future as well in order to see if there's a difference between the two. So why did we choose this topic? So me personally, I have a personal connection to this topic as exercise has been a big part of my life ever since 7th grade. And dancing became a part of my life in early high school when I started to gain an interest in it, but I didn't get to really express my enjoyment in it until I got to college. And then throughout the pandemic as well, I started to develop dance workouts as well, which actually proved to be a more enjoyable and also consistent workout plan that I was able to do throughout the pandemic. And so this personal connection and my recent workouts during the pandemic, that's what sort of gave me this interest in pursuing this topic further.
I have a similar background, but also slightly different from Paris's. So my interest also stems from my dance experience. I've been in recreational dance groups since I was about 3, although I did different sports throughout my childhood, once I came to college, I didn't really have a regular exercise regimen. I don't personally regularly go to the gym other than when I'm in dance group, and my dance group is my main form of exercise that I have at college. And because of that, I'm interested on how exercise can contribute to overall health and how exercise can be incentivized by enjoyment. Because whenever I've exercised, my primary goal is fun or social engagement more so than the actual health benefits of the exercise itself. So for me, it shows how important that enjoyment can be to incentivize exercising. And so that's why I was really interested in looking into dance as a form of exercise, because it's something that I've always enjoyed.
All right. Any questions? We're happy to take questions now.
I have a question. What were the specific 3 dances that you guys chose, or you have yet to decide that?
We have yet to decide that, but we're going to make sure that the difference in the music styles are unique enough so that we can get an accurate representation of which one is the most effective.
We're planning to look at the rhythm, the beats per minute of each song to try to vary dance style and intensity to see, obviously a very intense dance will burn a lot of calories, but at the same time, if a slightly less intense one is more enjoyable and might be more likely to follow through, then that one might be the one that we actually consider to be better.
Thank you. Also, just another quick question. Do you guys look at the difficulty of each song, like of each dance? Is that going into account of it?
Yes. So basically, in Just Dance, there's usually a rating of how difficult each one is. We're going to also account for that essentially when we design the study, when we do the study. I mean, so basically this ties into the whole principle of how the one that burns the most calories, the dance, isn't necessarily the one that's the most effective workout because it might not be as repeatable. And so that's where we're tying together the one that burns a lot of calories while also being enjoyable at the same time.
Great work. Thank you, guys.
Thanks. I see we have a question in the chat as well. Is the study looking for the most popular dances? Okay, so basically what we're doing is we're looking for which dance is the most repeatable while also burning it. It will also giving you a good workout while also burning a significant amount of calories. Yeah, I hope
so
that.
we're hoping to get like the exercise enjoyment results, like the PACES scale as we discussed, and then compare those enjoyment levels and the likelihood of the person repeating that form of exercise with how difficult they found it and how many calories they burnt, so that we can see like, is there like some sort of curve or is it more like linear? Like what is that kind of correlation between like difficulty and like calories burnt versus like enjoyment and like retention of the exercise plan?
Yeah.
Can you guys hear me?
Yeah, we can hear you.
Oh, okay, good. I'm sorry, I just wanted to make sure you can hear me. So I understand that you're trying to target obesity as well as mental health. So I was wondering, have you thought about motivation aspect of your study, and also how are you gonna provide incentive for your participants?
Yeah, that's a good question. So basically, the way we're going to sort of take into account the motivation aspect of it, this ties into how exercise isn't necessarily, people don't always find the motivation to keep up a consistent exercise routine. And so therefore, the motivation is going to sort of come in with that fun factor, the enjoyment factor that comes with doing with the dances themselves. And as to how we're going to provide incentives for that, we're going to, when we target our college students, we're going to essentially pretty much draw in demographics of students who are also interested in fitness, pretty much. Yeah. Ananya, do you have anything to add? Yeah, no problem.
Yeah, as for you asked about the mental health aspect with tying into motivation, and the initial study is mostly going to correlate like enjoyment factor and like that calories burnt, like that concrete number, but like that would be the initial investigation. But we have noted that like in future studies, depending on these results, it would be interesting to investigate if those calories burnt, if those physical benefits at that optimal level of exercise do correlate increased mental health benefits or not. But that's not something that we're specifically focusing on with this initial investigation.
Very interesting study. Thank you.
Anytime. Yeah, Ishan, do you have a question?
Yeah, I had a quick question, but also a comment. I really like this study. I think it's super important and definitely not talked about enough. So great job, guys. I really hope the best for you getting published and getting through IRB.
Thanks.
Yeah, there. So you said you're using one copy of Just Dance. Just as like a quick question, what platform do you hope to use it on? Like, I know Just Dance is on like every console. So is it like an Xbox 360 with Connect, Nintendo Switch, Nintendo Wii? What's your plan here?
So we haven't decided that one officially yet. I guess I'm leaning more towards using the Xbox Connect or the Nintendo Wii, as it encourages movement of the entire body pretty much, whereas one system essentially promotes more movement over the other, so that can give us a more accurate reading of the dance as a whole.
We're just going to make sure that we use the same platform, the same system, and like the same game each time, just so that we can like make sure it's standardized, because obviously like Just Dance 2014 on the Connect would have a different result than the same game on the Wii.
Mm-hmm. Well, that's awesome. Yeah, I wish you the best of luck.
Thank you.
Thank you.
Natalia?
Yeah, I completely agree with Ishan and also Dr. Pajuhish-Ganji. I think this is such an interesting study and I'm excited to see how it turns out. Thank you so much for sharing it with us. And I do have a quick question. So I read before that men tend to burn calories more than women, and I was wondering if you have considered comparing across gender as well, or if that could be something done later on. And also, I just wanted to say sorry that I turned off my camera, but I had some Wi-Fi connection issues, and I don't want it to affect the recording. So, yeah.
Yeah, it's all good. So yeah, we're going to, that's something we hope to examine in the future as well, like gender differences between essentially calories burned in a given amount of time for a workout. Like once we advance further with the study, we get potentially a different sample size with maybe a different demographic as well, then we can also think about the differences that would result from that as well.
Yeah, right now we're not focusing on comparing genders or any like specific really, but obviously like we're going to try to make sure that we get like a representative sample and then in the future perhaps investigate further.
Wonderful. Thank you so much.
Anytime. Any more questions?
All right, thanks for having us.
Yeah, thanks for listening.
Thank you. So I will introduce the next team, which is led by Dr. Pajuhesh Ganji, and the students are Anna Tentras, Nasser Ajay, and Daniela Tortosa. They also asked me to share a pre-recorded video with you all, so I'll be sharing my screen and we'll be playing their video.
Our research topic discusses the supporting cells of the central nervous system. Our focus as a research team, guided by Dr. Ganji, was to learn about the individual glial cells that support the central nervous system and how they interact with one another. The supporting cells we will focus on in this presentation are oligodendrocytes, microglia, and astrocytes. Since we could not meet in person, we each picked a glial cell and found articles about them. Meeting weekly, we would share our papers with the team and discuss what we learned. Over the past 6 months, we gained an understanding of where these glial cells originate from, their functions, and how they interact with one another and how they come together to support the central nervous system. We also delved into how these glial cells, when not functioning properly, can result in neurodegenerative diseases like multiple sclerosis, or MS. Jean Martin Charlotte was the first to describe the autoimmune mediated neurodegenerative disease, MS, in 1868. MS is characterized by the demyelination and oligodendrocyte loss of multiple focal points of focal sites through the central nervous system. The loss of myelin and oligodendrocytes is accompanied by local inflammation and infiltration of peripheral immune cells, which results in the interruption of axonal communications and functional deficits. Scientists are still unclear about whether oligodendrocyte death precedes inflammation or whether local inflammation drives oligodendrocyte loss. They also have not determined whether the loss of axonal communication is a consequence of demyelination or inflammation. Regardless of which event occurs first, demyelinated lesions, known as MS plaques, as seen as white patches in MRI scans, are the pathological hallmarks of MS. Due to the destruction of myelin, these lesions are littered across the brain of patients suffering from MS. The oligodendrocytes, which we will be discussing in more detail on the next slide, cannot keep up with the demand of remyelinating the lesions. This causes the progressive deterioration of the central nervous system, worsening the symptoms observed in MS over time. To fully understand MS, we first need to understand the functions of the supporting cells of the central nervous system, beginning with oligodendrocytes, which are derived from proliferative adult progenitor cells that give rise to oligodendrocyte progenitor cells, or OPCs, shown here. OPCs differentiate into pre-myelinating cells, and this differentiation is regulated by transcription factors, which are proteins that control the rate of transcription of genetic information. These pre-myelinating oligodendrocytes then mature into functioning myelinating oligodendrocytes to myelinate several axons or various regions of the same axon through the process of myelination. Myelination produces myelin sheath around the surrounding axons of the central nervous system. Here you can see an oligodendrocyte in green that is extending its processes to myelinate the several axons. Myelin is crucial when it comes to the conduction velocity of neural signals. Myelin insulates the axon, allowing the neuron to send signals rapidly through the central nervous system. The larger the diameter of an axon, the thicker the myelin is. Presence of myelin helps with keeping the ions close to the cell membrane and the axons, preserving the strength of signals as it travels down the axon. In this image, we are comparing an unmyelinated axon to a myelinated axon. The signal, which is the purple moving down the axon, travels much slower in an unmyelinated axon as compared to a myelinated axon. The gaps between myelin are called the nodes of Ranvier and contain clusters of ion channels responsible for maintaining a suitable amount of ions in the axon, which amplify the neural strength by allowing the signals to jump from one node to the next. This is known as saltatory conduction. If we closely look at a cross-section of a myelinated axon, we can see the individual myelin sheets surrounding an axon. These layers are made of phospholipids shown here in blue that are attached to one another by proteins such as MBP and PLP and MS. The oligodendrocytes may die, which can result in demyelination and interruption of neural signals.
Another supporting cell in the CNS are the microglia, which constitute approximately 5 to 20% of the total cellular population. They are the resident macrophages of the CNS and are derived from embryonic myeloid precursor cells in the yolk sac. Homeostatic microglia are critical for maintaining normal neuronal function, and in their non-pathological ramified state, microglia use long branch processes to survey their microenvironment for any assault. Upon the detection of injury or infection, microglia will shift to the activated phase with an amoeboid form and a larger soma. In this state, they behave as scavengers to clear out myelin debris, damaged proteins, and apoptotic cells from the CNS. Here, we can see how ramified and activated microglia look in actual tissues using IBA1 staining, which is a marker for microglia. Once activated, they can present neurodegenerative and pro-inflammatory characteristics that are known as M1 microglia releasing cytokines, which are small signaling proteins capable of activating other supporting cells in the CNS, such as A1 astrocytes. Microglia display significant heterogeneity in phenotype, and as the injury subsides, they can also have more M2 characteristics, releasing anti-inflammatory cytokines that may activate a different group of cells, such as A2 astrocytes, which are more neuroprotective. It has been shown that there are many more intermediate microglia phenotypes between M1 and M2 states. Microglia can phagocytize the myelin debris in multiple sclerosis.
Lastly, we'll talk about astrocytes. Astrocytes are derived from heterogeneous populations of progenitor cells in the neuroepithelium of the developing central nervous system. The cell body of an astrocyte spans 10 to 20 microns, and its processes radiate out for another 20 to 30 microns, occupying a huge amount of space in the central nervous system. There are two types of astrocytes in the central nervous system. The fibrous astrocytes have few organelles and long unbranched processes radiating from the central cell body. The long processes have vascular feet that encircle the capillaries present in the vicinity of the astrocytes. They are typically seen in the white matter of the brain and spinal cord. They are less common as compared to the protoplasmic astrocytes, which have short branch processes and abundant organelles in cytoplasm. Protoplasmic astrocytes are found more in gray matter of the brain and spinal cord. Next, we'll talk about the functions of astrocytes. Astrocytes are the most numerous cell types within the central nervous system and perform a variety of functions. They occupy a lot of space and can serve as a scaffold in the central nervous system. Here, you can see a specialized astrocyte in the retina called Mueller cell, which provides structural support to the retina and expands the entire thickness of the retina. Astrocytes are involved in glial scar tissue formation, as seen here in the spinal cord injury model. Upon injury, astrocytes shown in green proliferate and migrate towards the injury site. They then surround the area of injury and become activated, which result in hypertrophy of their processes, growing much larger, thicker, longer, and serve to wall off the area of injury, forming a scar, shown here in red. Another function of astrocytes is maintaining homostasis by constantly monitoring the interstitial fluid. They're either taking in ions or releasing ions to keep the ion concentrations constant. Perhaps one of the most important functions of astrocytes is forming the blood-brain barrier. Astrocytes prevent certain large molecules to pass through blood vessels. The end feet of astrocytes are plastered all over the blood vessels that are passing through the central nervous system. They prevent any leakage or unwanted substances from the blood into the capillaries. The astrocytic feet are seen here in green lining up the blood vessels. Lastly, astrocytes can clear neurotransmitters from synapses. Here you can see two axons, one green and one red, with a space between the two, which is the synapses. The blue astrocytes is clearing the excess neurotransmitters from the synapses and recycling it back to the green neuron. Upon injury, bidirectional communication between astrocytes and microglia modulates central nervous system inflammation through the secretion of multiple cytokines and inflammatory mediators. For instance, A1 astrocytes become activated by cytokines produced by M1 microglia and then can form a glial scar to prevent further neurodegenerative damage. Even though they do this to prevent further damage, it also hinders central nervous system recovery because some of the cytokines they produce can cause rapid cell death. On the other hand, A2 astrocytes are activated by M2 microglia and are involved in neural survival and tissue repair.
Multiple sclerosis research is still ongoing, and there are two main theories explaining how demyelination may occur in MS. The first one is the outside-in theory, which describes demyelination in the context of T-cell infiltration and microglial activation. In this model, the astrocytic feet that have lined up along the wall of blood vessels forming the blood-brain barrier are compromised, allowing immune cells to cross into the CNS. Antibody-mediated destruction to the myelin unit and activation of microglia occur via infiltrating immune cells. Destruction of myelin results in apoptosis in oligodendrocytes. In the inside-out theory, there can be different primary events leading to the recruitment of immune cells and myelin damage. In one model, microglial activation is the primary event, which can, similar to the previous model, cause myelin destruction and oligodendrocyte death. In the other model, oligopathy is the primary event, which assumes dysfunction of oligodendrocytes causes demyelination, which in turn leads to microglial activation and recruitment of immune cells to the CNS. There are compelling evidences for all these theories, making MS research intriguing and complicated. Here are our references. We thank you very much for your attention, and I'll be happy to answer any questions.
I just wanted to say that that was a very good presentation, and thank you so much for explaining all of that to us. I'm still thinking of a question, but as soon as I have one, I'll ask.
I have a question. Which of the different cell types do you think contribute most to like the disease pathogenesis?
I can kind of answer that. I personally really think it's a combination of all three because, as we kind of explained, each of them plays a different role in it. And I think like without the interactions between all of them, like MS wouldn't present the same way. So I really wouldn't be able to pick one. I would just definitely say just the way they all interact. I don't know, Nasser or Danny, if you have anything.
I'm thinking more of oligodendrocytes just due to the fact that they myelinate the axons, and when you have, oligodendrocyte loss and the loss of myelin, contributes to the symptoms we see in MS.
Yeah, I mean, I agree with Anna because all three play a role, and I think all three are significant. Even though astrocytes, they make up the majority of the brain and spinal cord, so maybe they all contribute, but like to different extents. So yeah.
Thank you. Does anybody else have any questions?
I actually have one question. So as you were researching about the supportive cells of the central nervous system in regards to multiple sclerosis, were you also looking at the treatment for multiple sclerosis, or were you able to find any current treatments or I guess like the most recent ones?
I know in the beginning of our research, we spent a little bit of time looking at it, but as we continued, we focused more on like the pathology of it and like all the roles of all the cells in it. So I'm not really able to speak on any current research for the medications, but I definitely know that there is a lot of research out there.
Thank you.
If nobody else has any questions, we can go on to the next team. I know it's supposed to start at 4:05, but if you guys are all here, team 5 is Xavier's team. You guys can share your screen and present.
Yeah, we can start now. Can everyone see it? Okay. Hi, I'm Xavier, and I'm here today with my team member Aram, and we'll be focusing on, well, we have focused on social deficits during our time with the symposium, and so we will just be introducing that and explaining some of the literature we've read explaining social deficits.
Hey guys, I'm Arham, and so basically what are social deficits? Social deficit is an inability or unwillingness to act in accordance to your age or physical condition and intelligence. And some examples include, but are not limited to, basic communication, empathy and intimacy, interpersonal skills, and then problem-solving skills along with accountability. And some common examples are autism spectrum disorder or ASD, attention deficit or hyperactivity disorder, and obsessive compulsive disorder. And the complexity of social deficits can be viewed in, they can be seen across many disorders, and each disorder has a different degree of which social deficits are expressed. So it all involves into a very complex area of study.
Okay, so we were interested in research that explores social deficits in varying disorders, how they're studied and researched, and the connection between an individual's genome and the expression of social deficits. So basically the biological aspect of the contribution of social deficits. So we had the honor of working with Dr. Liu, and Dr. Liu's research lab is currently exploring the correlation between neural circuit dynamics and social behavior for revealing the circuit level mechanism underlying social deficit. Recent research in her lab is focusing on measuring the cortical circuit function at single cell resolution in vivo during social interaction and learning tasks using mice models of ASD or autism spectrum disorder, and characterizing the abnormalities of neural circuit function and motor cortex that correlate with the development of movement deficiency in mouse models of ASD. So the long-term goal of Dr. Liu's lab is to use state-of-the-art in vivo calcium-2+ imaging technique, mouse genetics and optogenetics to characterize malfunction of neural circuit in mouse models of autism spectrum disorders, and to explore the possibility of intervention and rescue. So the purpose of Dr. Lu's lab and also the research that we looked at is to provide insight into the development of treatments for certain social deficits. So we didn't look at specifically the treatments themselves, but the different targets of treatment.
So the first piece of literature we looked at the default mode network, or the DMN, which is a brain network involved in understanding one's emotional state. And dysfunction or mutations to this can lead to social deficits that are found in autism. And this study compared resting state functional connectivity within individuals with ASD between the ages of 8 and 13 and a control group. And the researchers found that individuals on the spectrum displayed both over and under functional connectivity, both within the DMN and outside of it. And so basically that means that the DMN in ASD individuals shows poor segregation of connectivity. And this study confirms the potential for social deficits to be detected by the functional connection of the midline core. And this can be considered as an endophenotype. In addition to that, it can be concluded that the poor segregation of the DMN can lead to excitation and inhibition imbalance within individuals with ASD. The second study we looked at, this research paper revolved around the study of social neuroscience, as this paper relates to psychiatric disorders which are characterized by social deficits. The researchers used certain technologies such as human functional magnetic resonance imaging, or fMRI, to study how the brain and its neural circuits are modulated by analyzing neurotransmitter and receptor density in specific regions, and how disorders of social functioning such as autism may be caused by neural circuits failures to modulate these aspects. The researchers stated that in addition to possible structural issues with the brain regions, individuals with ASD may have abnormalities with dernic or GABAergic or serotonergic, or like consistent amount of different neurotransmitter signaling. Abnormalities mainly consist of serotonergic signaling, which has been found to be the main culprit, and with blood levels dropping down to in over 45% of autistic individuals. This is mainly caused by a uptake and degradation of monoamine, which is responsible for controlling serotonin blood levels in individuals with autism.
In the review article, Drug Discovery for Autism Spectrum Disorder, Challenges and Opportunities, the researchers provide a brief analysis of the current understanding of the pathophysiology, or the disordered physiological processes associated with disease or injury of ASD, which includes the most recent insights derived from the rapidly evolving genetics of ASD. So developing drugs for ASD has been challenging because of the limited understanding of its pathophysiology, difficulties in modeling the disease in vitro and in vivo, the heterogeneity of symptoms, and the lack of prior experience in clinical development. In the past few years, these challenges have been mitigated by considerable advances in our understanding of forms of ASD caused by single gene alterations, such as Fragile X syndrome, which is a genetic condition that causes a range of developmental problems, including learning disabilities and cognitive impairment, and tuberous sclerosis, which is a rare genetic condition that causes mainly benign tumors to develop in different parts of the body that most often affect the brain, skin, kidneys, heart, eyes, and lungs. These cases have allowed researchers to gain insights into the pathophysiological mechanisms underlying these conditions. In addition, they have aided in the development of animal models, mainly mouse models in most of our studies, and compounds with the potential for disease modification and clinical development. Moreover, genetic studies are illuminating the molecular pathophysiology of ASD, and new tools such as induced pluripotent stem cells offer new possibilities for drug screening and disease diagnostics. Finally, large-scale collaborations between academia and clinical industry are starting to address some of the key barriers to developing drugs for ASD. So researchers and clinicians alike are working together to overcome the barriers that are preventing the finding of drugs for ASD. in another review article titled The Genetic Landscapes of Autism Spectrum Disorders. They first observed the clinical and genetic heterogeneity of ASD, stating that beyond the unifying definition of ASD lies an extreme degree of clinical heterogeneity, ranging from profound to moderate impairment, so the symptoms of social deficits. Molecular and genetic studies have revealed that genetic landscape of ASD to be highly heterogeneous, with different types of genetic abnormalities located on almost all chromosomes with varying levels of penetrance. In recurrent de novo and inherited copy number variants in ASD, researchers found that recurrent chromosomal rearrangements are associated with ASD. The observation that deletions and duplications lead to different clinical outcomes indicates that a fine balance of correct gene dosage is necessary for human brain function and development. Further studies are warranted to identify the causative genes, or most likely the combination of causative genes, that increase the risk of ASD in individuals carrying these genomic rearrangements. It was seen that the genes associated with ASD are numerous and involved in multiple cellular functions, including chromatin remodeling, metabolism, mRNA translation, and synaptic function. Possible defects in homeostasis were concluded as consequences of mutations as well. Next, the researchers proposed that mouse models carrying mutations and pathways associated with ASD can display abnormalities in social interactions, communication, and repetitive behaviors, which is also seen in Dr. Liu's lab. And then the phenotypic variability highlighted in different models is reminiscent of the clinical heterogeneity observed in individuals with ASD. So again, the heterogeneity of social deficits that are expressed. So conclusion. So first, what we have learned. So me personally, we didn't really get a chance to really dive into the lab itself just because it's such a hands-on lab. And of course, we're virtual. So a lot of what we learned was through Dr. Liu describing what they do in her lab and presenting different research to us, having her colleagues present research to us as well. And then our reading and finding of articles that also relate to social deficits as well. So we mostly learned with that. And one thing I learned, well, I learned a lot of things, but the main thing I feel like I learned from this was that I think it's easy for people to assume that the cause of a lot of symptoms or even diseases themselves are all very direct and have a very obvious cause. But that's not the case at all with autism and related neurological disorders, as neural circuits are working together that lead to certain symptoms, and then also genes and their functions as well, and the heterogeneity of those. Aram, did you want to say anything about what you learned?
Yeah, the basic conclusions I got from my papers were that ASD and a lot of social deficits are impacted by hyperactivity or underactivity in certain neural circuits because everything is connected, and poor segregation of these connections can lead to ASD.
For plans for the future, since we were not able to go into the lab and really experience their research ourselves, hopefully with vaccines being distributed and the pandemic slowly but surely coming to a more stable condition, we'll be able to go and visit Dr. Liu's lab in the future and really witness firsthand the research that they're doing in the lab. Very excited to see the mouse models and yeah, learn more firsthand. So how will we incorporate what we have learned into our own goals and plans? My teammates and I, we joined this lab because we all have an interest or desire to learn more about autism. And from this, we were not only able to learn about different underlying mechanisms that lead to social deficits in autism, but also social deficits in other disorders as well. And with, I know one of my teammates is very interested in doing research with this later on. So it's just one way we can interpret and incorporate that into our future plans and goals.
And overall, we just wanted to thank AMPC and Dr. Liu for providing us help and a platform to display our research.
All right, thank you all for listening. And if you have any questions, we're here to answer them. if no one has any, what challenges do individuals with ASD face? So social deficits were our main, the main symptom we were looking at for ASD during our time with the symposium. So social deficits just refer to, like in the slides, like the lack of basic communication skills, a lack of accountability. I know a lot of them mainly affect their social interactions with other people. So that's the main thing we looked at. But social deficits incorporate a lot of other things like motor function as well. So it just depends on the individual too, because there are different, like people can display different social deficits, even if even if they're both diagnosed with ASD, the severity and extent to the expression of those symptoms can vary between people as well. I hope that answered your question.
I have another question, not related to the research, but maybe towards your experience with your research partners and the senior researcher. So with you guys, what do you feel that has been like the biggest takeaway from your time working this team? How have you guys like grown? And do you guys anticipate you might be working on the project in the future, or future anticipations for different projects?
Yeah, for sure. I feel like over the course of this virtual research, we all grew together because we had to, problem solving was a really big skill that we all took away from this, because we all have our own schedules and everything's virtual. We're all busy, and life is different as we all know it. And for sure, we talked with Dr. Liu, and we were very interested in her future research because we are up to date to it to some extent, and we all have an interest in it. So we would all like to join her in the future.
Thank you so much.
Yep, no problem.
Great presentation, by the way. Really loved listening.
Thank you.
Yeah, I don't have any questions, but I do want to say thank you to both of you. This was a great presentation, and it was a very interesting topic as well. And I also wanted to add a comment about a book called Look Me in the Eye. I'm not sure if you've heard of it before, but it's about this person with Asperger's, which is kind of similar to autism. So if you ever want to take a look at it to see some of the social deficits that the person himself describes about, it's very interesting as well. Yeah.
Thank you.
Yes, thank you. And thank you for everyone for listening. If there are any other questions, y'all can reach out to us personally, I guess, or send email, AMPC, you can answer them. But thank you all for listening.
Thank you both.
Thank you. Bye.
Thank you. So next up it's my symposium team led by Dr. Raul Chavez Valdez. My teammates weren't able to make it today, so we ended up pre-recording our video, and I'll be sharing my screen again with all of you, and I will be playing the recording.
My name is Marcel, and so happy to be here at the symposium. Here is our research topic, understanding the mechanisms of late neurodegeneration following perinatal stress. And the introductions are as follows.
Hi everyone, I will first introduce our primary researcher, Dr. Raul Chavez Valdez. He is an associate professor of pediatrics at the Johns Hopkins University School of Medicine, and his area of clinical expertise is neonatology. I am Natalia Beller, and I am a recent graduate from GW with a BS in cognitive neuroscience, and I am now a student at Columbia University's postbachelorate pre-medical program.
Again, hi everyone, my name is Marcel Carrasco, and I am a rising senior at George Washington University, also majoring in cognitive neuroscience, and I'm happy to be here.
Hi everyone, my name is Mahi Nawaz. I'm a rising sophomore at the George Washington University, majoring in neuroscience. The type of research we chose for our project was mostly literature review. Our main topic and focus throughout the project was to understand the effect of prenatal and postnatal stressors on the development of neurodegenerative disorders. We wanted to look into different stressors and how they impact the brain, which leads to different disorders. Through our research, it was found that Alzheimer's disease is the most common form of neurological dementia that affects 20 to 30 million people worldwide. Like many other neurological diseases, Alzheimer's disease does not yet have a quick cure or formative treatment that stops the progression of the disease. Most people who develop non-familiar Alzheimer's disease become symptomatic after age 60, while familiar Alzheimer's disease emerges earlier in life. The purpose of this review is to describe the potential links between prenatal and postnatal stressors and their interplay with genetic predisposition to increase the risk to develop Alzheimer's disease and other neurodegenerative disorders early in life.
Alrighty, bear with me, everyone. So the contents of our literature review, it's pretty hefty. It's a very long paper, not gonna lie. So to start off, as always, an introduction. And we went on to talk about 3 hits. The first hit was known as the genetic risk modifiers that Natalia went over. So we know that over our research that the most prominently known genetic risk modifier are the POE alleles, which I'm sure many of you have heard. Next, we went on to describing the second hit, which I researched, the in utero and perinatal stressors that contained just a few, like in utero malnutrition, maternal stress, perinatal asphyxia, and other things that you can see on the slide. And then lastly, the third hit, which Maheen researched, was composed of things such as TBIs, early life abuse, and neurotoxicants. During this research, we found that what was really interesting was that combined, we were able to correlate all of these early life hits to genetic risk factors. So kind of everything that goes on in life in the second and third hits can all be brought back onto the first hit, which are the genetic risk modifiers, which was really cool as we were researching, because we saw that a bunch of the things were linked, you know, which gave us a better idea onto seeing the development and the pathophysiology of Alzheimer's disease. Lastly, we researched the cellular and mechanistic hypothesis that are proven targets behind the development of Alzheimer's disease, making them the best way to discover treatments to finally, hopefully one day, cure Alzheimer's. And then our conclusion, of course.
So we first started our research in late November or early December. We started researching different scientific papers that could encompass the topic of our literature review. So we looked for reliable research that would focus on different genetic and perinatal stressors that lead to neurodegenerative disorders, as mentioned, with a focus on Alzheimer's disease. After we found the right papers, we decided to create a skeleton layout or a detailed outline so that we knew how to conduct our literature review in a clear and concise way. So we then began writing by dividing ourselves into the different sections that Marisabel mentioned. And we're still in the process of writing the literature review, almost finishing it. And our primary researcher is editing it as we go. So after we finish writing it, hopefully in about 15 days, we will be publishing it.
Through this research, we were able to learn many techniques. We learned how to conduct extensive literature research while finding reliable source and putting information from multiple scientific papers together. We have built our knowledge and deepened our understanding of neurodegenerative disorders with a focus on Alzheimer's disease and previous research and their limitations. We have also strengthened our teamwork, problem solving, and writing skills. And lastly, we have been able to discuss and apply our knowledge of this topic to different fields of medicine.
So in terms of our future plans and future research, we hope to first finish our first literature review on neurodegenerative disorders and perinatal stress. And after we do so, hopefully we get to start a new one, our second literature review, and this time focusing on neuropsychiatric disorders, such as depression or schizophrenia or even autism. And again, linking it to perinatal stress and genetic risk factors as well.
All right, well, thank you everyone for coming to this presentation. It's been a journey, but we couldn't have done it without each other. Definitely have been very lucky for AMPC to bring us all together, and we're here for any questions.
Thank you.
Thank you.
Yeah, so if anyone has any questions, please don't hesitate. Oh, Victoria, go ahead.
I have a question for you. So what was something surprising that you learned that, because I know you're like a cognitive neuroscience major, so you obviously learned so much, but was there something that you like learned that was like something that you had no idea about before that you thought was super interesting?
Yeah, I think that one thing that I should have thought about before, but I didn't, was the sex differences and how it might affect patients, females and males differently. And also they present the symptoms a little bit differently as well. So we found that amongst most of the genetic alleles or the genetic risk modifiers, there were some sexual dimorphisms or dimorphisms between gender, where women tend to be more affected than men. And this is something that I found pretty surprising because I wouldn't have thought about it beforehand.
Thank you. That's super interesting.
Yeah, if anyone else has any questions.
I actually, Nathalie, this is Joji. I have a question regarding the overall approach that you guys have taken, and this is not just unique to yours, but it does impact on what you've presented. The disease you're studying is so diverse in its origin and its manifestation. You've talked about the sexual dimorphism, but there are other aspects to it that makes it very differentially manifested based on the genetic mutations they have, environment that people have lived in. So the literature, and the second part is that this is not an under-researched disease. There's a ton of literature. So if you're thinking of getting into researching or reviewing a topic like that, do you think you've taken the right approach? And if you have, then what do you think was the right approach?
Well, in terms of researching, so we wanted to focus on perinatal stressors. And so as I imagine you know, that includes before, so the genetic risk factors, and then later on, the prenatal and postnatal stress factors. And so in order to assign most of the known stressors to these different stages, we thought that our researcher really emphasized the importance of relating back to both the gender differences, which is very prominent, especially in regards to the genetic factors, and also the environmental factors, as we see mostly later on. So it's mostly when in regards to the prenatal and the postnatal states. And so with basis on that and seeing how these states might lead one to the other, we felt that that would be an important approach for us to conduct. But if you have any advices for us, any suggestions of conducting it differently, I would be happy to hear it.
Well, that's the thing with research. Each way of doing it is the right way of doing it. It's just a matter of how much longer it takes and what your journey brings you to. There is no good way to tour a place. You can tour a place any which way you want, and you will get with different things. And that's why your perspective matters than mine or anyone else's. You've taken the journey, and that's what matters, what you think.
Thank you. If anyone has any other questions, I'm happy to answer them. But if not, then we can move along.
Does anybody else have any questions? Okay, so the next team is actually my team. So Amy and Presley, those are my research partners. Amy, you can share your screen now.
Yeah.
Can you guys see that?
It's still loading. Yeah, there it is. My name is Victoria, and I'll be presenting with Amy and Presley. Oh wait, go back. I just wanted to say thank you to Jody, James, and Brian for working with us every week on this project. I've learned so much, and we're super excited to talk about all the work we've done over these past few months on the biological pathways that contribute to the severity of heart disease in Duchenne muscular dystrophy models. So our research project focuses on Duchenne muscular dystrophy, which is a severe muscle wasting disease resulting from mutations in the X-linked dystrophin gene that affects boys and mildly affects female genetic carriers. The image below shows the dystrophin glycoprotein complex. Loss of dystrophin, you can see it in the purple, that's the dystrophin gene, and the dystrophin protein. This protein provides the muscles with structural support. And when you lose this, it leads to damage, inflammation, and fibrosis in skeletal and heart muscles. Affected boys experience symptoms like cardiac dysfunction, which also manifests in female carriers. Recent studies for DMD have extended life expectancy in boys. However, this has led to increased incidences of cardiomyopathy. Increased muscle damage leads to the activation of fibrotic pathways and stiffens the extracellular matrix, which provides support to cells, and also results in calcification. In our study, we aim to investigate the biological processes that contribute to heart tissue damage in the more severe DMD mouse model D2-MDX as compared to the milder model Black 10-MDX.
Sorry, my network went out. I'm so sorry. So this figure deals with the progression of DMD within the cardiac tissue. We can see the severity in affected patients in regards to the progression of cardiomyopathy. The first image is a normal heart, and through DMD, there's like mild fibrosis in the heart tissue, which is like during the later late teens. And eventually there's like a severe fibrosis in the early 20s, which eventually leads to the heart becoming enlarged and leading to heart failure. The image below displays a more focused view of a tissue which is stained to show the fibrosis that is present. In the next slide, there's like a comparison between B10-MDX and D2-MDX mice. B10-MDX demonstrates less severe progression of the disease with little or mild late onset of cardiomyopathy. The D2-MDX mice shows a severe progression and an early onset of fibrosis and calcification. And we are interested in finding why the DM2-MDX is more severe than the B10-MDX model.
So moving on to the methodology, to examine the data more closely, we used RNA sequencing to assess changes in gene expression at the overall genome level. So we performed RNA sequencing on the heart muscle tissue from 2 DMD mouse models so that we could investigate changes in gene expression at the disease onset. And after that, we used bioinformatics to compare the RNA sequencing data to identify all genes that were differentially expressed between the mild and severe models, and then perform functional analysis using databases like gene ontology to identify the most significant pathways contributing to the severity of cardiac disease in DMD. And most recently, what we did was we used String, which is a tool that allows us to determine the gene interactions, both known and predicted, to identify which genes are most prominent within the extracellular matrix and immune response pathways. And then to further identify the gene groupings, we conducted 2 quality control analyses. The first of the 2 things we did was a Pca analysis. This helped us to determine how the samples are grouped together at the gene expression level, and also showed us that the samples within each group do actually cluster together. And then after we conducted the Pca analysis, we were able to analyze its results and found that a heat map would be helpful in further validating and visualizing the results we got from the Pca. So here in the heat map, we used the top 500 most significantly altered genes of our data sets that were sorted based on a 0.5 adjusted p-value ranking followed by a fold change ordering. And you can see in the Pca that the top 3 samples are from the B10 models, and the bottom 4 samples are from the D2-MDX models. But overall, the heat map shows the relative expression of these top genes so that we can better assess the overall pattern of gene expression in each mouse type. And so by using some of the gene ontology databases, we found the most significantly altered processes to be that of the extracellular matrix, followed by the immune and inflammatory response pathways. So in the image in the right, you can see that the bigger the statistical significance, the more crucial role it plays in having an impact on the progression of DMD if it is altered. And we were able to derive this chart through data collected in our database, or through the database Gorilla, which is a database that allows us to analyze ranked lists of genes. And you can see here that the inflammatory or immune response pathways are shown in the red bars, whereas the extracellular matrix, which I'll shorten to ECM from here on out, is shown with the blue bars. And then if you look to the left side of the screen, this is from the database String, which is, again, a functional analysis tool, which helps us to gain better visualization of the gene associations. So here, as you can see in the ECM string, these are ECM-related pathway genes that are prominent in cardiomyopathy and the progression of DMD. It shows the top 28 genes we believe to be the most significant that are clustered from a much larger pool of gene ontology genes. And underneath it, the immune and inflammatory related genes show the top 24 most significant genes that we found. And these genes were selected based on cell type. And then in cross-referencing our gene database with literature, we were able to essentially derive lists of relevant genes in the ECM and immune response pathways. And this allowed us to see that the most significant of these genes were generally the ones that held the highest fold change, as this meant that they're the most upregulated and as a result will contribute to the progression of DMD to a greater extent.
We also use another functional analysis tool called gprofiler to validate the results of our Gorilla analysis. We use many functional analysis tools, but these were the ones that we thought gave the most clear information about the different pathways. So we identified specific pathways associated with inflammation and ECM remodeling that were significantly altered at disease onset in the severe D2-MDX hearts that should contribute to fibrosis and cardiomyopathy. In this image, you can see that the higher up the dot, the more significant the process is. The top two are in the blue, which is related to extracellular matrix, and the bottom four are related to immune and inflammatory pathways. So progressive infiltration of immune cells such as neutrophils, macrophages, T cells, and B cells in response to muscle cell damage leads to activation of fibrotic pathways, which results in a stiffened ECM and further tissue damage and muscle degeneration.
So next, what we did was we conducted downstream analysis. Within the extracellular matrix, the most important cell types were things like elastin or collagen, fibronectin, or TGF-beta. And so for the ECM, we decided to focus on TGF-beta. And when the TGF-beta is altered, an upregulation leads us to conclude that the increase in tissue deposition in type 1 collagen of fibrosis is due to an increase in these gene transcription rates. And the signal transduction pathways show the mechanisms involved in the transcription of these type 1 collagen genes by TGF-beta, where the two distinct pathways are created, because as you can see in the top row, the type 1 collagen are SMAD gene dependent, whereas the bottom row with the TIMP1 gene is found in the cultured fibroblasts. The SMAD gene is included in the upregulation leading to collagen synthesis and is responsible for the transactivation because it's brought in by the TGF-beta. But ultimately, this is just one of the many possible ways a change in the expression of a gene can lead to the degradation of the extracellular matrix and ultimately fibrosis, which then increases the severity of DMD.
In terms of a specific immune response pathway that is altered in our data set, I saw that toll-like receptor 4, TREM1, and SYK were all dysregulated in the D2 model as compared to the black 10 model. TREM1 at the top, you can see it up here, interacts with toll-like receptor 4 to induce cytokine production, which heightens the immune response. And then downstream, SYK binds to LTR4 and is activated and subsequently activates other genes as well as the J and K pathway, which results in greater cytoskeleton rearrangement, reactive oxygen species generation, and cytokine secretion in macrophages, which is all greater in the D2 model as compared to the black 10. These results and the dysregulation of this pathway in the D2 model proposes a mechanism for cardiac tissue fibrosis and an explanation for the severity of cardiomyopathy in the D2 model. Additionally, the lack of dystrophin weakens cardiomyocytes, and this damage results in cardiomyopathy. Chronic and prolonged damage induces immune responses, which leads to inflammation and immune cell infiltration. This prolonged infiltration by immune cells, such as T cells, B cells, neutrophils, and macrophages, eventually leads to fibrosis. You can see them in the second chart. In particular, I did a deep dive into macrophage-related genes and how the infiltration of macrophages affects the cardiac tissue and results in fibrosis. The following genes, CCL2, MSR1, ARG1, and SOCH3 are immune/macrophage-related genes and are upregulated in the more severe D2 model, and thus provide possible mechanisms to explain the severity of disease in the D2 model. For example, CCL2 and SOCH3 have effects on the JAK-STAT pathway, and upregulation of this pathway causes increased inflammation, which leads to cardiac deficits. In fact, SOCH3 knockout mice are known to prevent myocardial apoptosis and sustain activation of cardioprotective signaling molecules, which can mean that SOCH3 is a possible therapeutic target for improving cardiac deficits in the D2 model. Additionally, MSR1 upregulation activates J and K and induces a pro-inflammatory state, while ARG1 is a widely used macrophage marker where upregulation leads to tissue fibrosis. All of these genes are involved in the immune response system and are known macrophage-related genes that lead to greater inflammation and ultimately cardiac tissue fibrosis, which was seen in the pathology image that you saw before, and explains why there's so many cardiac deficits and issues with cardiac function. We discussed the immune and ECM pathways separately, but they do work together to produce the cardiac pathology of inflammation and fibrosis that you've seen. On the left side, you can see that cardiac tissue damage activates macrophages and fibroblasts, which leads to maladaptive cardiac remodeling. Macrophages are immune related and fibroblasts are ECM related, but these two together cause the remodeling, which has to do with fibrosis and ultimately leads to cardiac dysfunction and cardiomyopathy, which is what we've been trying to explain in the D2 model. And then on the right side, this model shows that there are different like ECM gene families and cell types that Amy touched upon, like collagens, TIFs, and MMPs, and how combined with inflammation cause cardiac deficits and fibrosis and result in the pathology in the more severe D2 mouse model due to dystrophin deficiency. So our next steps in our investigation are to validate the predictions we've made based on the RNA sequencing data that we've analyzed. We will do some wet lab evaluations. So for the extracellular matrix, we plan on doing trichrome staining and Western blot to identify collagen deposition in the cardiac tissue. And then in terms of the immune cell types, we've identified we'll be doing immunostaining to see the density of T cells, B cells, and neutrophils and macrophages in the cardiac tissue. And finally, we can also do qPCR to validate expression of the dysregulated genes that we discussed earlier.
Thank you for your time.
Yeah, thank you guys so much for listening and coming out today to the first symposium. We were all very excited about it. And I've had a great time working on this research, and would just like to thank again Jody and James and Brian for helping us out so much and meeting with us every week to help us on this project. I've learned definitely like so much over these past few months.
Thank you, guys.
So I have a question. This is Luba Kaldurik. I'm sorry, I'm not raising my hand, but do you know if two strains, B10 and D2, exhibit differences in inflammatory response in other diseases, or you didn't come across that information in your evaluation of these pathways?
So our study was focused only on Duchenne muscular dystrophy, and that is like all the literature we've been reading up on. And so I don't think I can answer that question fully, since that was like our main muscular dystrophy is the focus of our study. So we haven't like looked at other diseases that use these models, but that can be like a future investigation for us.
Yeah, but initially we were looking at skeletal muscles in relation to muscular dystrophy, but the focus of like the presentation in our most current research is based on cardiac muscles and how they're impacted by the progression of DMD.
That's fair enough. Thank you.
Thank you. Ishan, do you have a question?
Yeah, you guys definitely did so well in this. I think this is such a great presentation and it shows your dedication throughout this process. So congratulations. I know
Thank you.
with this as well. I think DMD and cardiomyopathy is a really interesting topic. And I just wanted your opinion on like your greatest challenge through this virtual research process.
I would definitely say just coming together and trying to draw connections between our pathways, because especially since we are virtual, it was really hard to like have a cohesive thought, I guess you can say, because what we did is for the most part, we came up with different pathways. So I would be doing the extracellular matrix, for example, and then Victoria would be doing the immune response. And then what we would have to do is like collaborate to try and find overlapping genes between the pathways, because ultimately that would be what is most important, or like supposedly or predictably the most significant in affecting DMD and cardiomyopathy.
Yeah, I would say definitely integrating like separate pathways that I found were most like prominent or upregulated or downregulated that would affect the D2 models and cause it to be more severe. Like integrating them together into one cohesive like idea is definitely difficult because there are so many like different genes that can have effects, and it's really about like just making predictions based on the regulation and the dysregulation of the genes. And so I'm honestly still working on trying to create like a cohesive model, and definitely connecting the immune and ECM as well to explain the severity of the disease in D2 as compared to Black 10.
Yeah, we're still far from done.
Thank you for your answer. Yeah, you guys are doing amazing. I'll reiterate my thank you for sharing, and yeah, great job.
Thanks, Ishan. If nobody else has any other questions, we can move on to the next group. Let me know if you guys have any questions, and then we can move forward. All right, so the next team is team 8, and this is Dylan's team. So if you would like to share your screen.
Hi, everybody. My name is Dylan Allen Dogan, and I am a rising junior studying neuroscience, and I'm part of the team that was focusing on aberrant expression of Urea Cycle genes in cancer. The other members of our team include Melissa Owusu-Ansah, who is also a rising junior majoring in neuroscience, Nikki Guptan, who is a rising sophomore majoring in neuroscience, and the leader of our research team was Dr. Kaldovic, who is part of the GW Department of Genomics and Precision Medicine. Now, before we get into the brunt of our research, I think we need to establish a very basic understanding of the urea cycle. The urea cycle takes place primarily in the liver and to a lesser extent in the kidneys. And its primary function is to produce urea. The importance of urea is that it helps take out excess ammonia that accumulates inside organisms. And this is really important because if there is an excess accumulation of urea, this can lead to hyperammonemia, and excess of ammonia. It can lead to hyperammonemia, and this can lead to all different kinds of bad prognoses for the organism. Our research started out with 8 genes, part of the Urea Cycle, and then we narrowed it down to 3 main ones that we focused on. Those include CPS1, NAGS, and citrin. Now, CPS1, this gene, when it is expressed on tissues where it normally is not expressed, it leads to an excess of carbonyl phosphate, which leads to increased pyridine synthesis and cancer cell proliferation. The second gene that we focused on was citrin. And when citrin is overexpressed, excess aspartate accumulates, and this leads to nucleotide synthesis and cancer cell proliferation again. And the third gene we focused on was NAGS. And we found that NAGS was necessary for CPS1 expression and its activity. So theoretically, we found that they should have similar expression patterns in similar tissues, as well as their variants.
We then also looked at past research in exploring how all the urea cycle genes are expressed in tissues. And so what we have here are diagrams that show like a primary site or an organ, for example, the liver, kidney, and heart. And on the y-axis, we have FPKM, which is a measure of how much protein or mRNA is present in of a gene in a specific organ or tissue. And so what we found was that NAGS, CPS1, OTC, and arginase were all expressed in the liver, and with arginase, it is only expressed in the liver. And also looking at the spectrum, so we also noticed was that OTC, ASS1, and ASL have much broader spectrums, which indicates that they're also highly expressed in more tissues. And what we also found interesting was that NAGS, OTC, and CPS1 are also highly expressed in the intestine. And so just keeping in mind that NAGS, OTC, and CPS1 are highly expressed in the liver and the intestine, we also looked to how they're expressed in cancers as well. And so what we have over here is a heat map. As you can see, we have all the 8 genes of the urea cycle and how they're expressed in cancers. And so basically how this heat map was generated was, well, the basis for the heat map was we're comparing the tissue, the cancerous tissue, to the normal surrounding tissue. So like for the red bars, for example, under CHOL, so like because it's red, it means that the gene is underexpressed relative to the normal tissue that is surrounded by, that surrounds it. And so for the green, it would be like the gene is expressed in higher levels than in the normal surrounding tissue. And so what we also found interesting was that NAGS, CPS1, and OTC were also like overexpressed in other tissues that weren't the liver or the intestine. For example, we have GBM, which is an acronym for glioblastoma, which occurs in the brain. They're actually all but one of the 8 genes were overexpressed in that area. So that was also an interesting point for us. And so we later just focused on how these how the expressions and variants in cancers compared to like what was previously found in just patient data. And so just revisiting that from the previous heat map, we also saw that a lot of the pattern expression for NAGS and CPS1 did have a small overlap. And so just going back to, which is an expected pattern because NAGS does contribute to CPS1 expression as previously mentioned, and a defect in any one of the urea cycle genes can also lead to hyperammonemia as also previously mentioned, and which is a very bad thing because it leads to a slew of health problems and can also lead to more serious issues like brain damage and coma.
So next, we wanted to collect sequence variations in order to compare them. And so we collected them from 5 different sources. The first one we collected from literature, which is where just patient variants are reported. Next, we have LOVD, which is a publicly available database
of patient mutations, and then ClinVar, which is a database of variants that are detected in people that are sequenced for diagnostic purposes, meaning that these people are not quote unquote healthy. Next, we have Nomad, which is a database of variants that are found in healthy people across the world. And then we also collected data from Cosmic, which is a database of cancer mutations. And so essentially what we did was we compared sequence variations found in cancer patients versus healthy people. And next we put them into Venn diagrams. And what we found was that most sequence variations that are found in cancer are unique to cancers themselves, meaning that they're not shared with variants that are common variants that are found in other people. And we can see that in how the Cosmic database variants are unique to themselves. We can also see from the Venn diagram that there's an overlap between literature variants as well as LVD, because all of those are patient mutations. And there's also an overlap between Nomad and patient mutations, because it's known that each person carries about 100 deleterious disease-causing sequence variations. However, for Urea Cycle disorders, you need 2 copies, except for OTC disorders. So most people will be manifested as healthy. So in conclusion, we can see that, as I stated before, most sequence variants found in cancers are unique to cancers themselves. And so our next direction with what we found is to look at what type of mutations are present in cancers, whether they're things like amino acid mutations or mutations that have to do with splicing and things like that. We also want to look at what enhances the activity of specifically NAGS, OTC, and CPS1 activity in cancer as well. And finally, we want to thank Dr. Keldovic for working with us on this project and teaching us a lot about the urea cycle itself and sort of its broader implications. And then we also want to thank AMPC for connecting us to Dr. Keldovic and making this project possible. So thank you. If anyone has any questions, we'll be happy to answer them.
This is Jody. I have 2 questions actually. One was, do you think that the entire urea cycle is active in those cancer cells?
So we found that that they are active to a certain extent. Some of them are underexpressed and some of them are overexpressed. So our next step is actually just to see specifically what those genes are. And right now we're focusing on those 3, but we plan on later taking a look at those other genes that we mentioned and we showed in the overexpression data.
But that speaks to what genes are up and down, but they need to work in a concert for the entire urea cycle to work, right?
Correct, yeah.
So do you think that that's happening, that the entire urea cycle is operational?
It is operational, but some of the genes, so what we found that CPS1 needs, for example, CPS1 needs NAGS in order to function, but we're seeing that in some cancers, NAGS isn't present. So we're trying to figure out, in cancer cells, is NAGS necessary for CPS1 activity or is it not? So just to give an example, Melissa and Dylan want to add on.
Yeah, I think it's mostly functional, but just not as efficient as it would be in a normally. So basically the urea cycle does turn ammonia into urea. So and a common issue with urea cycle disorders is hyperammonemia. And so because that's an issue, it indicates that the urea cycle isn't functioning as it should be because there's an excess of ammonia. So that may point us to think that maybe it's not it is functioning, just not as efficient as it should be. But we've seen that it does function somehow.
Thank you. And the second question I had was, what do you think is causing these genes to start expressing in these tumors? Have you looked into that, or from your genome analysis, do you have any thoughts? What might be the reason why it is so strongly suppressed in other cells in the body, but suddenly in these tumors they are starting to express these, specifically these urea cycle genes?
I think it's because, well, we haven't really gone into detail for that part yet, but a lot of the genes that we've seen help with pyrimidine biosynthesis, which helps with cancer cell proliferation. So we're thinking that might be one of the reasons why that these genes are overexpressed in some of the tumor cells.
Thank you. And just a final comment. This is a really interesting way of looking at things, what you guys have done. And it looks like it's a lot of work that you guys have put together to learn all these different approaches. So congratulations. This is wonderful.
Thank you.
Thank you.
Does anybody else have any questions? All right. Well, thank you so much, guys, for presenting. Very interesting topic. To move on to the next group, they could not be here today. There was only one person who was supposed to be presenting. However, they had an emergency. So I do have a little note from them about what their research was about, and hopefully maybe in the future they can present at a different symposium. So I'll read their note. In our research, we wanted to better understand the differences between human and mouse retinal ganglion cells. Retinal ganglion cells are a cluster of neurons located on the retina of the eye. They receive visual information from photoreceptors through two intermediate neuron types. Based on the data we collected, we observed that human cells are bigger than mouse cells. Normally, bigger cells have more elaborate and longer dendrite branches, which was not the case here, probably because mice were sacrificed, which means that the cell membranes were better preserved and that the fluorescent dye sank in well and gave better and accurate results. So that was what their research was about. I'm sure they have more to work on. But I know the next and final presentation of today isn't supposed to start until 5:20. I'm not sure if you guys are here. This is Parissa's group. If not, we can wait until 5:20 for the last presentation. But if you guys are here and are ready to present, we can also do that. So just let me know. All right, let me see.
Yeah, I don't see Parissa Samia, but I do see Dalila just joined us. So just to explain it to you, I don't know if you heard all of everything that we said. The group that was supposed to be presenting right now wasn't able to join us today because of some family emergencies, but we were just wondering if you guys would like to begin at your scheduled times or if you're all ready to begin. I believe that we're still waiting for Parissa, but we have one more student here, and so we can just wait for her and begin.
Okay, so if we're gonna wait for Parissa to come in, you guys want to take a break, get a snack, something to drink. Oh, okay, here she is. Yeah, so just let us know if you want to present now or wait until 5:20. It's up to you guys.
We can go now.
Okay. All right, the floor is yours. You can share your screen. Thank you guys.
Okay. All right.
Okay, so this is me, Parissa, and Samia's group about neurofibromatosis type 1. Sorry about this, a long name. Yeah. So for introduction, Parissa is, we're all freshmen. Parissa is sophomores now. Praise is a biology major on the pre-med track. Samia is a neuroscience and psychology major on the pre-med track. And I am a cognitive neuroscience major also on the pre-med track. All women in STEM.
Okay. Okay, so the disease here in question is neurofibromatosis type 1. It's incredibly variable in its clinical presentation, and no two patients will have the exact same case. It affects multiple organ systems, and it's chronic in nature. It's inherited through genetics as it's caused by mutation of the synonymous NF1 gene, which is located on chromosome 17. Some of the most recognizable symptoms are the neurofibromas and plexiform neurofibromas, which are tumors found in the peripheral nervous system nerves and can get pretty out of control to a really tough level. So to begin our work, Samia, Dalila, and I all met with our PI, Dr. Tomczyk, who recommended some beginning literature for us to get acquainted with the disease and its neuroscientific pathways. We then conducted an additional self-driven literature review to gain more depth on specific topics, which we will be covering in a bit. Some of the hardest parts of the readings were found in the complicated cellular pathways, so we used additional resources to try to make more sense of it. We then began a draft of our literature review to discuss some of the topics we found interesting, like clinical symptomatology, the cognitive profile, and the cellular and genetic pathways involved. Animal models were something our PI advised us to look into, as this is where most of the future of neurofibromatosis research lies. NF1 pathology is difficult, and so we do still have some room for improvement in our understandings, and a lot of the research surrounding NF1 have their own knowledge gaps as well that we have to navigate since this disease is largely not well understood. We did, however, go into the treatments available for NF1 patients, which was really interesting and actually relatively quite recent. And so the three topics we want to review with you all today are cellular pathways, the cognitive profile and treatments, and the various animal models of NF1. Sammy's going to get us started off, so I'll hand it over to her.
All right, so first of all, regarding the cellular pathways, as Parisa mentioned earlier in the presentation, NF1 is genetically inherited through a mutation in the NF1 gene, which is located on the 17th chromosome. The two main cellular pathways that we were interested in looking at with regards to NF1 is the RAS-RAF-MEK-ERT pathway. So this is an extracellular regulated protein kinase pathway, a MAPK pathway, commonly referred to. And there is a potential connection to cancer, which makes sense regarding the fact that the hallmark symptom that many NF1 patients experience are plexiform neurofibromas located on peripheral nerves throughout the PNS. In addition to this, there is a supplemental signaling pathway that is also involved, which is the PI3-AKT-mTOR pathway, and this has to do with cell proliferation. And below here, there is a picture just demonstrating how different ligands and cellular mechanisms are involved regarding ATP and synthesis and things like that that allow for either the uncontrollable cell growth that produces plexiform neurofibromas or the proliferation of cells. And for the cognitive profile and treatment, so essentially the cognitive profile within NF1 patients ranges drastically in the presentation. And as a result, there are a multitude of learning disabilities and IQ issues that a lot of NF1 patients run into. There is a significantly higher likelihood of experiencing ADD or ADHD in comparison to control patients. A remarkable thing that we found also is difficulties with language processing, memory retention, and of course, attention and concentration. Currently, in the current literature, there is substantial evidence that shows that surgery, radiation, and pain management are the top three, treatment protocols involved in treating NF1, considering there is no current cure. The reason why radiation and pain management are so prominent within current pain management strategies is because of the fact that plexiform neurofibromas located on the peripheral nerves induce a lot of pain, and as a result is a chief complaint among NF1 patients. However, recent advancements in NF1 research through either invertebrate models or vertebral models, such as mini pigs or mice, have allowed for a kinase inhibitor known as Koselugo to be FDA approved for use in patients with inoperable plexiform neurofibromas, which are essentially just like other plexiform neurofibromas. However, either their location or the recurrence of the plexiform neurofibroma renders them inoperable.
So one of the animal models that were mentioned were mini pigs, and this was really exciting because mini pigs or swine aren't necessarily seen as a model that is used a lot. And they have been added to the research into NF1 because they are a better choice than mice or Drosophila because of how similar they are to humans. Their nervous, cardiovascular, integumentary systems are all similar to those of humans, and they also have recurrent nonsense mutation that is also mimicked in humans with NF1. And it was found in mini pigs with NF1. They had multiple dark brown skin patches, which were similar to CALMS in human patients, which just provides more research into NF1 and helps us understand more about the disease. Parisa will talk about Drosophila models now.
Yeah, all right. So the Drosophila melanogaster model has been used to study certain pathways involved in neurofibromatosis as well. The DNF1 gene is highly conserved and is 55% identical to the human NF1 gene, allowing for studies to manipulate the ortholog and use Drosophila as a model system for research purposes. One such study on Drosophila used DNA transposons, which can be used as gene editing vectors, so that NF1 genes were rendered inert in the genetic sequence of the experimental group. When a Pavlovian olfactory behavioral paradigm was used, flies lacking the NF1 gene were demonstrated a decrease in cognitive performance. Notably, the displayed impairments were in olfactory learning and long-term memory. Reduced growth during development and reduced overall adult body size were observed, as were failures in circadian rhythm and dysfunction in mitochondria. In another study, in important activating ligand for DALK, the secreted protein, which is called jelly belly and is also known as JEB, was found in the mushroom bodies of Drosophila brains, which are a pretty important brain structure for memory and learning. DALK and DNF1 have been known to colocalize, and DALK is an upstream regulator of DNF1. ALK, which is short for anaplastic lymphoma kinase, is an RTK protein found in mammals. A parallel pathway also exists in Drosophila, which has been of great interest to researchers as inhibition of the DALK gene represents a potential future treatment of NF1, this being the rationale for the targeted manipulation of DALK. Mushroom bodies are critical for olfactory learning and memory, where DRK, an adapter protein necessary in RAS signaling, is also commonly used. When cross-sections of brains were stained with anti-DALK antibodies, the most staining was found to be present in these mushroom body calyces. And the increased staining of mushroom body calyces indicated a high level of mRNA expression of the DALK gene in the associated areas. Increased DALK expression is associated with an increased likelihood of learning deficits, and it acts as a limiting negative regulator of olfactory-associated learning. Regulation of the larval fly cell size is regulated by both the DALK and JEB genes that are understood to influence growth, but neither gene seems to have a direct effect on cell proliferation.
So for this project, we are all currently working on a literature review of NF1 pathophysiology and pathways, and we are hoping to publish in the future, possibly in the GW undergraduate review. Academically and professionally, we all hope to graduate with a bachelor's degree by 2024 and hope to go to medical school afterwards. Yeah, thank you.
Thank you guys so much for your presentation. If anybody has any questions about their presentation, their project, please ask them now. I actually have a question. So if you could do wet lab research on this topic, would you choose to do it on the pig model or the Drosophila model?
That's a really good question. I think personally, probably the mini pigs, just because I looked into mini pigs more, and there's not a lot of research on mini pigs yet with diseases. And I think being on that cutting edge, like with the NF1 and using mini pigs as a model would just be more beneficial and more like interesting. But Drosophila would be cool too, but I just feel like mini pigs would be where I would go.
I definitely agree. I feel like there's just like so much potential for new discoveries just because of how close the mini pig brain is to the human brain. So I think it would be like really fascinating to be on that kind of, side of things for sure.
Even though I agree with Parisa and Dalila, I would probably go ahead with the Drosophila model only because there was a certain mechanism of the RAS-RAF-MEK-ERT signaling pathway that I was a bit confused about regarding like an EGRF mechanism that possibly leads to either increased cell growth or proliferation. And because I didn't fully understand it due to the limitations in research, that's probably something that I would be interested in exploring further.
Thank you guys so much. And if you guys want to just touch on like what you guys learned from working with each other through like the virtual environment and your other future plans, if you are planning on maybe doing wet lab research on this topic or anything like that.
I think, well, things are harder virtually, but I think the group, me, Parisa, and Samia, like it was a really good group. I love both of them so much. And it was fun working with them and like struggling understanding some stuff with them and just learning from that struggle, like bouncing off things with each other. It was really beneficial and it was really fun. And maybe we can do a wet lab in the future. Maybe if like after the literature review, we might like get into contact with our PI, like, hey, can we do more research on this? But yeah.
Yeah, definitely. It was such a great experience, like going through all of it and trying to figure out what works best in terms of like task coordination and time management and really like coming together as a team to get everything done. So that was great to like develop those kind of skills. And even in like a virtual environment, like it really did feel like it was coming close to real research just with how intense we were with the actual subjects and how in depth we got on all the literature. And yeah, definitely, it would be great to have an opportunity to work in a wet lab once COVID dies down and everything goes back to normal a little bit. But for right now, we're just working on the literature review, which is cool.
Yeah, I really enjoyed working with both Parisa and Dalila. The fact that our PI gave us a lot of room to explore different cognitive mechanisms, cellular signaling pathways, it gave us a steep learning curve in the beginning, especially because none of us had really known what NF1 was prior to working with Dr. Tomczyk. But I think now NF1 is an extremely interesting clinical disease, and I look forward to either doing more research throughout the literature review or possibly going into wet lab research with both Dalila and Parisa. So yeah.
Thank you guys so much. Does anybody else have any questions for them?
I actually just wanted to make a comment to Victoria's question and the answer you guys gave with regards to choosing the model. One of the things, if you guys do end up doing wet lab research with it, is worth keeping in mind. The question and the models need to be aligned. If you're doing a discovery-based approach, pig is going to be a very difficult model to work with because it's just very unwieldy. But if you're doing human relevant research with regards to pathology or anatomical aspects, then Drosophila is going to be a very difficult thing to work with. So it's important to keep in mind the reason there are hundreds of different models of different sorts of things is because they're good for something, not good for other things. And your question should drive it rather than the overall relevance or closeness to a human system.
Okay, thank you. That's definitely something worth considering. Thank you very much. That's good advice.
Really interesting project, guys. Congratulations.
Well, if nobody else has any questions for them, they were the last group to present. I'd just like to thank everybody for coming out today and watching everybody present. And thank you everybody so much for all of your hard work over these past months working on your projects. And yeah, AMPC is lucky to have such great members who are so dedicated to science and research. And we're excited to see what the research symposium can hold in the future. And if anybody else has anything they want to say or comment on about like the symposium process or anything like that, feel free. We're always happy to hear feedback about it and to improve in the future. And I'd just like to thank everybody again.
Yeah, thank you everyone for coming out today. We really appreciate it. All of you are absolutely amazing and you did wonderful jobs throughout. So congratulations to you. And I hope you have the best of luck in furthering your own research. And also make sure to come to tomorrow's symposium as well, because it's not going to stop. We just got more amazing people coming in. So yeah, just support your fellow AMPC students. Thank you.
Thank you.